Okay, good morning students. In previous class under the chapter Cell, the unit of life, in that particular chapter we have discussed regarding the organelles like mitochondria, the structure of mitochondria, endoplasmic reticulum, etc. In today's class we are going to start with the next organelle. Those are the plasmids. Plasmids are found in all the plants and euglenites. We are going to find these plasmids in all the plants and euglenites. And based upon the pigments which are present in a plastids have the specific pigments. Plastids have the specific pigment. Based upon the type of pigments, plastids can be classified as. This is very important. First thing is what I told you. Plastids, these are the organelles, these are also the membrane bound organelles, and these plastids are present in all the plant cells and euglenites. All the plant cells and euglenites. Okay. Next point is these plastids are consist of a specific pigments, and based upon those specific pigments, these are classified as what are those different types of plastids. Based upon the specific types of pigments are of three types. One is chloroplast, second one is chromoplast, third one is leucoplast. What I told you? Chloroplast, chromoplast and leucoplast. And what else you are going to remember now? What are the different pigments are there? Okay. So in chloroplast you are going to find two pigments. Okay. We are going to find the two pigments. What are those? One is chlorophyll and another one is carotenoids. Chlorophyll and carotenoids. In the chromoplast, these chromoplasts are the fat soluble plastids. And these are having the two pigments. Those are carotene and xanthophylls. These are like a carotenoids. Carotene and xanthophylls. And next one is leucoplast. This is colorless in the sense leucoplasts are the plastids which are colorless plastids, and these are meant for the store nutrients, for the storage of nutrients, or they are going to store the nutrients. And what are the different pigments which are present in the leucoplasts are amyloplasts, elaioplasts and aryloclasts. Okay, now these are what the pigments which are specifically present in a different type of plastids, in the sense chloroplast, chromoplast and leucoplast. ಅರ್ಥ ಆಯ್ತಾ? ಏನಪ್ಪ ಅಂದ್ರೆ plastids chloroplast, chromoplast and leucoplast. These are the three different kinds of plastids based upon the presence of specific pigment. ಅಥವಾ specific pigment ಇರ್ತಾವೆ. Chloroplast ದಾಗ ಯಾವ pigment ಇರ್ತಾವೆ? Chlorophyll ಮತ್ತು carotenoids. Chromophylls ದಾಗ ಇರ್ತಾವೆ carotene and xanthophylls. Leucoplast ದಾಗ ಇರ್ತಾವಪ್ಪ ಅಂದ್ರೆ amyloplasts, elaioplasts and aryloclasts. Okay. So what is the function of these chloroplasts? Okay. First, chloroplast function. These whatever the pigments are there. What are those pigments? Chlorophyll and carotenoids. These pigments are going to trapping the light energy. Trapping ಅಂದ್ರೆ ಹಿಡಿಯೋದು. Trapping the light energy for the photosynthesis. Photosynthesis ಆಗಬೇಕಾದ್ರೆ ಏನೊಂದು ಲೈಟ್ ಏನು ನೀವು ಅಂತಿರುತ್ತೆ ಸೂರ್ಯನ ಬರುವಂತ ಕಿರಣ ಹಿಡಿದಿಡಬೇಕಾಗ್ತೈತಿ. ಅದಕ್ಕೆ ಉದ್ದಿ ಸಂಶ್ಲೇಷಣ ಕ್ರಿಯೆಗಾಗಿ that is done by these chlorophyll and carotenoids. That is what I written here, trapping of the light energy for the photosynthesis. Next, these carotene and xanthophylls which are present in a chromoplast, what they used to do? They give the part of the plant yellow, orange or red coloration. In the sense, it is going to give the coloration for the different parts of a plant. That should be yellow or yellow, orange or red color. Next, I told you leucoplasts. These are containing these three water enzymes, and what they used to help in the sense they are helpful for the storage of nutrients. In the sense, amyloplasts it store the carbohydrates like starch, example potato. Elaioplast that is going to store the oils and fats. Aryloclast that is going to store the proteins. These all are very important as a point of your competitive exam. Simply you are not supposed to go and take out the A, B, C, D like that. from first year onwards you used to start for preparing for the competitive exam. Okay, whatever I told you, these are the what the different types of a plasmids based upon the specific pigments, and you have to remember which pigment is present in a chloroplast, what are the pigments are present in a chromoplast, what are the pigments are present in a leucoplast, and afterwards what is the function of those pigments in the particular plastids. In the sense, chlorophyll A and carotenoids trapping the light energy for the photosynthesis. And I will tell you, based upon this concept, whatever I written on the board is 10% specific pigment based upon the plasmid different types, and I given the example and all. These all are present in your NCERT book, and based upon this much only the concept, even the concept minimum 10 question तैयार मारेगा. How many? 10 questions needs to prepare for the what need or a CET. Understood? So you have to make clear that what are the different types of pigments, different types of plastids based upon the specific pigment. Then what are the pigments are present in a chloroplast? What are the pigments are present in a chromoplast? And what are the pigments are present in a leucoplast? And what is the function of those? What is the function of those? And what are means name the plasmid? Which one of the plasmid means plastid which is fat soluble? That time fat soluble plastids are chromoplast. So likewise they are going to prepare the questions for the NEET or a CET. And for 1 mark 2 mark question they used to ask you these things. Name the pigments present in a chloroplast, name the pigments present in a chromoplast, name the pigment present in a leucoplast. What is the function of chlorophyll and carotenoids? That is the trapping the light energy for the photosynthesis. Likewise we used to prepare for the answering the questions. Okay, this is about what the plastids which are differentiate or the different kinds of plastids based upon the specific pigments present in a plastids. Now next we are going to study the structure of this chloroplast. We are going to study the structure of chloroplast. The majority of these chloroplasts of the green plants are found in a mesophyll cells of a leaves. Okay, these chloroplasts we are going to study in detail about the structure of this chloroplast in this particular chapter. The chloroplasts are present in all the green plants, but in which part of the green plant they are going to present? The majority of the chloroplasts of a green plants are found in mesophyll cells of a leaves. Which cells of the leaves? Mesophyll cells of the leaves. This is also very important for 1 mark question. Majority of the chloroplasts of the green plants are found in a mesophyll leaves of the mesophyll cells of the leaves. Okay. So this is about what first point regarding the chloroplast. Now we are going to start discussing regarding the structure of chloroplast. We are going to start discussing the structure of chloroplast. Okay, in structure of chloroplast, what else you are going to remember in the sense? Likewise, we were studied regarding the mitochondria. Likewise, chloroplast also what bounded with the two membranes. One is outer membrane, whatever I draw outside, and another one is a inner membrane. Outer membrane and inner membrane. Next one is this inner membrane is less permeable. Less permeable in the sense what? This is going to allow some molecules or some materials pass into the what we are going to call as a chloroplast, and outside the chloroplast they are going to permeate less materials or the molecule. That's what here it is a less permeable. Then likewise the mitochondria, here you are going to get the stromal laminae, granules, stroma and thallopoids. So what is the explanation regarding the chloroplast? The chloroplasts are the lens-shaped oval or a spherical discoid ribbon-like organelles. These are lens-shaped oval, spherical or discoid ribbon-like organelles And the measurement of this, in the sense, this is what we are going to call as the length. This is the width. Length of the chloroplasts are usually 5 to 10 micrometers, and weight of the chloroplasts are usually 2 to 4 micrometers. And what is going to happen? The number of these chloroplasts are varies from varies from 1 chloroplast per cell in a Chlamydomonas. This is very important. This is a blue green algae. Chlamydomonas is a green algae. Chlamydomonas is a green algae. In Chlamydomonas green algae, you are going to find only 1 chloroplast per cell. One cell daga just one chloroplast irtೈತಿ. ಅಲ್ಲಿಂದ ನೀವು next 20 to 14 mesophyll cells higher plants na ಬಂದ್ರೆ 20 to 40 ಒಂದು mesophyll cells ನಾಗ ಏನಂತಪ್ಪ ಅಂದ್ರೆ chloroplast ಇರ್ತಾವ. ಅಂದ್ರೆ the number varies from 1 per cell in a Chlamydomonas to the 20 to 40 in mesophyll cells. This is about the number regarding the chloroplasts which are present in Chlamydomonas to the higher plants. Afterwards, the number of organized flattened membranes, flattened membranous sacs. Yes, 1, 2, 3. These are the flattened membranous sacs are arranged in a starch. We are going to call them as a thalappoids. Okay, what I told you, the space in the inner membrane is called as a stroma. This is the inner membrane, and in between the inner membrane you are going to have this space. This space, complete space, we are going to call as a stroma, and in stroma you are going to get what? The number of organized flattened membranous sacs. Number of flattened organized sacs, those are called as a thalappoids. Those are called as a thalappoids. And in thalappoids, see 1, 2, 3, 4. These all are this structure, compound structure. This complete structure is called as a thalappoid. Then thalappoids are arranged in a stalk-like points. Here, these are the stalk-like points. 1, 2, 3, 4. These are the stalk-like points, and those are called as a granule. Those are called as a granule. Granule ಅಂತಂದ್ರೆ ಏನು? ಈ thalappoid ಅಂದ್ರೆ ಇದೆಲ್ಲ complete thalappoid ಇರುತ್ತೆ. This is complete thalappoid. Okay, and these thalappoid, in thalappoid you are going to get point-like structures which are arranged. Point-like structures which are arranged. Those point-like structures are called as a granule. Granule is a singular, and that is grana. What we are going to call it as a plural. So here I written thalappoids are arranged in a stalk-like points are called as a grana, and intergranal thalappoids, or we are going to call that one as a intergranal thalappoids. Understood? Then these flat-like structures, whatever the thalappoids are there, they are joined with each other. They are joined with each other, and that is named as stroma lamellae. That is tubular structures called as a stromal lamellae. This much you are going to remember about the chloroplast. Chloroplast has a two membrane, outer membrane and inner membrane. Inner membrane is less permeable. Inside the inner membrane you are going to get the space called as a stroma, and that stroma consists of arranged things that is called as thalappoids. In thalappoids you are going to find point-like structures called as a granules, and these thalappoids are arranged, means attached with each other with the help of stroma lamellae. Along with these, the stroma consists of double-stranded DNA and ribosomes. Double-stranded DNA and ribosomes. What I told you, along with the earlier whatever I explained the structure, there is a chloroplast containing the double-stranded DNA and ribosomes, and these ribosomes are of 70S. Then also this stroma containing the enzymes. Those are helpful for the synthesis of carbohydrates and proteins. And last one is chlorophyll pigments are present on a thalappoids. Pigments are present on a thalappoids. The thalappoids in chloroplast, on which you are going to find the pigments. In chloroplast, the pigments are present on a thalappoids. The pigments are present on a thalappoids. This is very important. Okay, so this is regarding what plastids. In plastids, we have discussed the different types of plastids based upon the specific pigments. Based upon the specific pigments, there are three kinds: chloroplast, chromoplast, and leucoplast. And we have discussed here in detail the structure of chloroplast and its function. Okay, this is what regarding the plastids. This is what regarding the plastids. Okay, the question is going to ask you people, draw a neat label diagram. Explain the structure of chloroplast. Explain the structure of chloroplast. Or on the basis of specific pigments, what are the different types of a plastics? What are the different types of a plastics? Or they are going to ask you, name the pigments which are present in a chloroplast, name the pigments which are present in a chromoplast, name the pigments which are present in a leucoplast. Likewise, the questions are going to arise for the final examination. Okay, so this is about what we have studied regarding the plastics and all. Now, okay, now we are going to study the next organelle, that is ribosomes. Okay, still for so far, whatever the organelles we are studying up to the plastics, chloroplast, all those are a membrane bound organelles. Now, the next organelle we are going to study, that is the ribosomes. Ribosomes are the non-membrane bound organelles. Ribosomes are the non-membrane bound organelles, and they are playing an important role in a protein synthesis. It is you know about this. Now we are going to study what are the ribosomes. These ribosomes are the granular structures which are present in a cytoplasm, and each ribosomes are made up of a two subunits. Ribosomes are made up of a two subunits. Okay. Whenever they are found in a two subunits. So ribosomes are the granular structures which are observed by George Pallade in 1953 under the electron microscope. Who is observed first time? The ribosomes. Ribosomes are observed first time George Pallade in 1953 under the electron microscope. And they are composed of, in ribosomes, what else you are going to find? The ribosomes are composed of RNA and proteins. RNA and proteins. In what ribosomes are composed of RNA and proteins, but they are not surrounded by any membrane. In the sense, it is a non-membrane bound organelle. Ribosomes are the non-membrane bound organelles. In the sense, what they are composed of RNA and proteins. Okay, RNA and proteins. Next, and they do not surrounded by any membrane. They do not surrounded by any membrane. And I told you the ribosomes are present in both the cells. In the sense, eukaryotic cells and prokaryotic cells. If they are present in eukaryotic cell, they are of about 80S, and in prokaryotic cells, ribosomes are 70S. And I written here S. This S is represents what? Sedberg's unit. This is represent as a Sedberg unit, and this Sedberg unit is used for the measurement. Indirectly is used to measure the density and size of these ribosomes. Understood? Density and size of these ribosomes. This much you are going to remember regarding the ribosomes. Ribosomes are the granular structures which are observed first time by the George Pallade in 1953 under the electron microscope, and these are composed of proteins and RNAs. They do not have any what? Membrane. They do not have any membranes surrounding it. Then these two are present in both eukaryotic and prokaryotic cells. In eukaryotic cell they are 80S, and in prokaryotic cell they are 70S. S is represent the Sedberg unit, and this Sedberg unit is used for indirectly measurement of a density and size of the ribosomes. Okay, this is regarding what next organelle, ribosomes. Okay, so in today's class, what else we have discussed? In today's class, we have discussed about the plastics, the different types of plastics based upon the specific pigments. Then we have studied in detail the structure of chloroplast. Then another one organelle we have studied regarding the ribosomes, and these ribosomes are the granular structures first observed by the George Pallade, and I told you these are composed of RNA and protein. Then these are present in both the prokaryotic and eukaryotes, and Sedberg unit, yes, represent the Sedberg unit, and that is used indirectly for the measurement of density and size. So this is about the today's class. I will continue next with the next organelle. I'm going to stop here. Thank you.